What we are going to do here is have a little discussion or an interview where I would be the interviewee and I'm the interviewer mostly about the future, because Arvid is very interested in the future and is a very active researcher still. So we will come to the current project at the end of this presentation. But we will, since most of you are probably not familiar with this field, we will also give you a brief history how all this came to happen. Psychopharmacology that we will discuss today is a discipline that is quite young. It all started in the 50s, one could say, and then it has now gone on for 60 years, more or less. And it's an interesting discipline because, on the one hand, and I think you would agree, we really don't know very much about the brain. Our ignorance is much, much larger than our knowledge. But on the other hand, with those fragments of knowledge that we do have, it has been possible to improve the lives of billions of people. And in the lead of this development, and the definitely figure for all this development has been Arvid, and he has been in this field from its start until now, and will be so also for the forthcoming years. So here we have a person who can give a picture of the development of psychopharmacology from where it started. And you'll also have some example of course, of Arvid's major discoveries in this field that have been definitely pivotal for the progress of the entire field. But I would like to start, Arvid, with exactly what Bengt referred to, and that is with this. This is a book called The War of the Soups and the Sparks, i.e. when you entered research, there was in fact a war going on. And here are two of the major soldiers in that war, Otto Lewy and John Eccles. Could you please, what was this war all about?
Yeah, well, Otto Lewy you mentioned. He was the one who really provided the first proof that the signal from a nerve to the next organ, whatever, in the body, is not necessarily an electrical signal, but can be a chemical. And he did some ingenious experiments in the early 20s on the frog heart, isolated, actually on two isolated frog hearts. So that was the start. There had been some speculations, very smart jobs actually before, but no proof. But Otto Lewy, in the frog heart, he made it. But then, of course, others said, all right, this is the frog, but that cannot be the same in the mammals. So then came a lot of important people. Of course, well, I couldn't name or mention all the names, but there were a couple of people who, there was one fellow who was who really managed, together with his colleagues, to prove that there is actually chemicals being released even in mammals, in different organs, even from nerve to muscle, which the others thought was impossible because of everything is so quick, there's so much speed, so it has to be an electrical signal. So these were the two, and you mentioned them.
And then you went to about the brain, the reluctance was even higher, and then John Eccles was one of the fighters until the bitter end, wasn't he?
Of course, absolutely. He was in favor of the spark.
Of the sparks, yes. So we have the soup guy to the left and the spark guy to the right here.
That's correct. Yes. But Lewy, he was not involved in the fight.
No. Okay. No, he was the fundamental.
He came before. He came before. Yeah, but the fight between the sparks and the soup was, of course, much lower than the sparks, and it remained like that for a long time. Yeah. The soup people weren't really any good scientists, you know. It was the sparks who did it.
Yeah.
And so that went on, and it was some very famous fights in London where the people got together. And very, very well attended, and a lot of fights.
And you were there?
No, this was
not
before
this
my
was before.
time.
Okay,
This
but then
was
we have a second fight that we'll come back to later.
Oh,
the second
that will
fight
be
in
later.
London.
Yes,
we'll
yes.
come back to that. But okay,
Yes.
because you joined this fight when before it was even. Was it all over when you joined? I will just go on with a few slides here. This is just for those not at all involved in neuroscience, and I will just, what we are talking about here is of course the connection between one nerve and the next, the synapse or the synapse, as you see it here. And the soup people believed in chemical transmission here, and the spark people believed that there were some tiny fibers mediating electrical stimulation. And this is when you, let's come back to your debut in this. This is, you went to the United States in 1955 to visit Bernard Brodie. And here I've tried to illustrate how you are going back with the flight to Lund to carry on the work after having spent half a year in NIH. Please tell us about this stay.
I had been working on in a very different field, an exciting one, using something very modern at that time, radioactive isotopes, to study the skeleton and the growth of the skeleton in rats. But the pharmacologists who were my superiors, they felt in the long run that was not a good thing for a pharmacologist to do. So I thought I have to switch. And thanks to some good friends, I happened to come to a very fabulous lab headed by Bernard B. Brodie. And that was one of the really a mecca of modern chemical pharmacology. It was exactly the place I wanted to get to. So I was very fortunate. And he brought me right into the middle of something very exciting. They had just made the discovery that one of the antipsychotic drugs that were used at that time, reserpine, if you inject that into animals and then analyze for a chemical, fairly newly discovered chemical that occurred also in the brain, serotonin. If you inject this compound to animals, serotonin disappears from the brain and from other organs in the body. So that was what happened. I was very lucky. I came there in the right moment. And they put me right on to this to do some in vitro work to prove that this particular molecule, reserpine, does indeed act directly on the storage of serotonin in blood platelets. So that was how it started.
We have a lot of young future researchers here. Is it important to be at the right place at the right time?
Yes, that is terribly important. Yes.
And how do you see to that?
Well, you must have some superior up in the sky to help you to do that, yes.
Okay, so you went back after 1 year to Lund and carried on the work with these amines in the brain. And before going on to what you did actually find, I would just show you this slide. When I started many years later at your department, and this was kind of a holy grail at the department. The aminco was in the center of everything. And that leads me to a question about methodology. How important are methods for development, do you think, in your field? And how important was the aminco?
Enormously important. Actually, when I worked on the skeleton, then it was also happened to be an entirely new technique. Measurements of, well, first the radioactive isotopes had become available, and then there was equipment to measure it. And now, in this particular lab, in Dr. Brodie's lab, there was the first prototype of this fabulous instrument, which was a spectrophotofluorometer, by means of which, for the first time, one could measure very low amounts of compounds, even such small amounts like serotonin. These mysterious compounds that we had that were in our tissues in extremely low, in micrograms per gram, that kind of thing, very low. So, but for the first time, they could be measured chemically. There were some bioassays that were available before. So I was enormously fortunate to be right in again, to be there when the first prototype of this particular instrument was there. And I think I was perhaps the one that got more mileage than anybody else from this particular instrument.
I think so. You bought one when you come back. You had funding. You bought one, and that was the start of the.
Yes.
Of your, this is a enormously important experiment, of course. What is happening here?
Well, Brody and his colleagues, they were so excited about serotonin. So, but I had, coming from a more physiological background, physiological pharmacology, I knew about other compounds that also occurred in the brains, and a bit different chemically, but after all rather similar. They are called monoamines, these things. And I thought perhaps we should take a look at these other chemicals. And one of them was adrenaline. And then there was another one, noradrenaline, that was one of the compounds that had been found to be a neurotransmitter in the peripheral nervous system. And because it was only in the peripheral nervous system that chemical transmission was at all seriously considered at that time. So in any event, I thought we should measure noradrenaline. And so we did that, and we gave reserpine. And that was back in Lund. So this was in Lund, and together with some colleagues, one of them a histochemic, a very clever histochemist, Hillap, who was also already in the field of adrenaline. So we made experiments with giving reserpine to animals, and we measured noradrenaline and found it also came down. And then when we went on further, we found that actually the reason why reserpine could do things to animals had nothing to do with either serotonin or noradrenaline. I mean, the most important thing was another compound called dopamine that at that time was considered to be just a precursor, not interested, without any physiological function, according to the bioassays they were doing with the gut and so forth in those days. So I came to we and my colleagues and I came to the conclusion, it's dopamine. So, and what we see here is what is actually, you have the animals on top there, the rabbits. They are not very wake, they can't move very well, and their wakefulness is very low. And if we now give, and that is because they have been given this antipsychotic drug, reserpine. But then what we did, we tried to refill the stalls of the monoamines, or especially then catecholamine, by means of a precursor. But we couldn't give the compound itself, we couldn't get into the brain. So we gave a precursor called dopa, L-dopa. And those animals, we see the same animals at the bottom here. They had been pre-treated with reserpine, and then they got an injection of dopa, and within IV injection, within 15 minutes, they were up and running, very dramatic.
You must have been amazed.
Yes, that was astonishing,
a miracle.
Yes. And of course, we thought at that time, we hadn't penetrated this problem so much. We thought it was noradrenaline coming back, because that was what had disappeared. But we found it wasn't noradrenaline. These animals that are up and running still have very little noradrenaline in their brains. But what we found, dopamine. All of a sudden, dopamine, this precursor, became a very important compound. And that was really a spearhead, I can say, in the future development that led to the concept of chemical transmission in the brain also, not only in the peripheral system.
And now we are coming back. I will go forward with some questions regarding this also, but just go back to the London meetings. Now we have the second London meeting, because now you have realized that you are a strong believer in chemical transmission in brain, and the amines playing a part here. And then you went to a London meeting.
Yeah, and that meeting on adrenergic mechanisms, that was the same kind of symposium. That was the dominating person there was one of the Nobel laureates, Sir Henry Dale, and he was actually the one who was fighting with John Eccles that we talked about earlier. And he was there, he was the boss, and nobody would object to what he said. He was 85 years old, a little bit younger than I am now. But he was a very strong person. So when he had heard our story, we reported about this fabulous experiment. They came to the conclusion that this is a serious mistake. We misunderstood it altogether. We were looking at the toxic effect of L-dopa. It had nothing to do with any neurotransmission or whatever. So we have to go back we were really dismissed altogether. And everybody agreed we were all wrong.
But you were not discouraged by this experience.
On the other hand, I felt very excited because we knew we were right. There was no problem. On the other, it was very good because at that time, since almost everybody who was in the field was at that meeting, and all of them agreed we were wrong, and some of them had done some work before in the field, so they couldn't, but now they agreed we were all wrong. So therefore, when it finally came out that we were right, they couldn't come and say, look, I did this before you. So it was a very successful meeting for me, this London meeting.
Okay. I must ask you one thing that always amazed me with this experiment and amazed me until today. And that is, as we will discuss a little more later, the chemical transmission, there is a chemical transmission in brain. And that is extremely regulated in an extreme sophisticated way with feedback mechanisms, autoreceptors, different kinds of electric firing, regulating the release of the transmitters, and so on and so forth. Isn't it amazing that an exogenously administered transmitter could restore a subtle thing, a complicated thing like locomotion, given that this should be so exactly regulated?
Yes, I agree. And for example, when we look at other neurotransmitters that later on came into the field, for example, if we go back to serotonin, if you do similar experiments with serotonin, you give 5-hydroxytryptophan and so forth, which is a precursor of serotonin, and what do you see? Very complicated things, but it so happened that with dopamine is so. Even though dopamine is involved in very complex neurocircuitries, when it comes to movement, it so happened that you could do this kind of experiment. You could take away dopamine, and the animal was down, all the way down. You could refill dopamine, that the animal came up. It's very fortunate, very unusual. And again,
you were lucky again.
I was lucky, yes.
Okay, this is just the two pivotal papers here, one in Nature where you demonstrate that dopa could reverse locomotion inhibition by reserpine, and then one where you claim that dopamine is present in brain and a transmitter. Just a brief comment, these were published in Nature and Science, but after that you have never been interested in impact factors or journals. A lot of young research today learn that to get somewhere you must publish in the snobbish journals. You have never cared about that. Did you care about that in the beginning of your career?
Well, in those days, Nature and Science were not so enormously prestigious. So it seemed to be natural to send them. It was not because it was Nature or Science or something very important. It was just a couple of journals that were around to send.
Okay.
Now, it's very different, of course.
And since then, you have never been impressed by that aspect?
No, I think it's rather one should be careful. If you have something that you know is something very new, very important, you should avoid going to one of the big prestigious journals. Put it in a rather obscure journal, because then you have published, you have it, you have the priority, and nobody notices, so you can go ahead and start to understand what you really have found, you know.
And this... And we shall see an example of that. You did that with a very important
there
finding.
are some examples. Yes,
Okay.
absolutely.
Okay. And let's now go to the clinical implications of this finding. Of course, all of you know about Parkinson's disease, where you have this reduced locomotion functioning and where you have a dopamine depletion in brain. And of course, the DOPA was, as a consequence of Arvid's experiment, DOPA was introduced as a treatment for Parkinson, and today, 50 years later, DOPA remains first line of treatment for this disorder. And obviously, millions of patients have been enormously helped by this. Did you foresee that consequence when you saw the rabbits?
Not certainly. When we published the effect, the rabbit data that you saw, we didn't even understand it can have anything with Parkinson's disease to do. It's rather when we later on found that dopamine occurs especially in those parts of the brain, in the so-called basal ganglia, that were supposed to be very important in connection with movements. And also other pieces of the puzzle, I shouldn't go into details, that made us understand that, of course, what we are looking at is a kind of a model of Parkinson's disease. So that's how it came about. But of course, when all these pieces of the puzzle got together, we thought we had something very important.
Yeah. Okay. Let me ask you a question on principle. You are from a medical faculty, you're an MD. Medical research have two purposes, finding the truth and helping patients. And this obviously was basic research that led to a very immediate benefit for numerous patients. And today, many people argue that we, in fact, know very little about brain function, and billions and millions, for example, of EU money are now spent on trying to really understand the brain by computer systems and stuff like that, more or less realistic plans, I would say. On the other hand, we have had research more directed towards the immediate help of patients, that perhaps you don't need to understand everything, you can go directly to help people. And there are still, of course, unmet medical need in psychiatry and neurology. How would you make priorities between these two? If you have a lot of money, should they go to really understand the function of the brain, or should one keep on with pharmacological research aiming to improve functioning in the short term?
Yes, well, of course, I have been fortunate in the sense that it so happened that even though we were working in really basal research, basic research, we happened to be very close to an application to the bedside. So from that point on, I have been very fortunate. But later on, of course, I have understood, we're looking at how different lines of research are ongoing, that what is called translational research is not priority. Actually, I think many scientists feel in this field that they feel that this is not high quality research. This is so close to the bedside. It should be something more, dramatic, that perhaps in 100 years from now could become something important. So therefore, I think there is too little emphasis on translational research, and the one basic research that aims more directly to treatment. But I think now some initiative here in Sweden now seems to realize that clinical research that should be basic but still aim at bedside benefit should be emphasized more. So I think we are moving a little bit slowly in the right direction.
While EU has gone for big science in neuroscience, they spend billions of dollars on big science instead.
Yes. And of course that nowadays it's so, if you have, it looks as if the goal in science is to have a paper published in one of the big journals. Then the champagne is up. And nobody thinks about what is this any good for the patients? That is a later thing.
Okay, so I think your current work also illustrates that now you are very focused on the patient. In fact, we are coming back to that shortly. But so in the end of your career, you were mostly, it was by accident you came in towards the bedside. But now you are, this is some of your incentives for.
Oh, sure. Yeah, the more I got in touch with the bedside,
okay,
the more I started to be a believer in translational research, or even though I started out from basic research.
A brief note about another disorder. This is two gentlemen in France, two famous psychiatrists, especially Delay was a very famous psychiatrist in the early 50s, and they just by accident came to give a drug called chlorpromazine to schizophrenic patients. This was a drug that has been used as a drug for inducing relaxation before operations, and then some surgeons suggested it should be tested in psychiatry, and they found that it had a dramatic effect on the hallucinations, the thought disorders characterizing schizophrenia, and it changed the entire face of psychiatry for a decade, for the forthcoming decade. A lot of people could go back to the society from the hospital and so on and so forth. And still, this is a kind of drugs that are being used for schizophrenia and other psychosis. No one knew at the time how they work. This is one of the many accidental findings that have characterized psychiatry and psychopharmacology. But a few years later, this was one of the findings that you hid in an quite not very well-known journal, and it was in fact ignored or neglected for many years. Tell us about it.
That is the most cited journal or article of mine.
Today it is.
From Acta Pharmacologica, a very obscure journal.
But it was ignored for the first 10 years or so.
Oh, yes.
The citation started after 10 years.
Yes, it was ignored. And what happened was that we could identify an effect for the first time of chlorpromazine that made sense, that could explain the various pharmacological and clinical actions of chlorpromazine. And that was an effect on receptors for monoamines, for especially dopamine, but also noradrenaline. And actually, we mentioned also as a possibility serotonin might be involved. So, and that, from starting out from that, we came, well, of course, something in between here. Before we came to this model, we must mention the histochemist work also. And this colleague of mine, Hillarp, who was from the very outset after I arrived back from Brody's lab, that by means of histochemical work one could really identify these amines in nerve terminals and in nerve cell bodies that already immediately make one understand we are dealing with neurotransmitters. So that confirmed the whole thing. And
in fact, if you see at the background of these slides, here you have a typical Hillarp.
Yeah, that's correct, yes.
Yeah, okay.
So we could put up, that was in 1964, for the first time a model that it's a complicated model. But the circle, the big circle that you see here, is a transection of a nerve terminal. It's a nerve terminal that could store a monoamine, either dopamine, noradrenaline, or reserpine. Not a mixture of them, but one of them, either one of them. And within that circle, you have two small bodies. One is a storage granules or vesicles, what you call it, that stores the monoamine in question, and the other, a monoamine oxidase, is another one that's in the mitochondria, of course, and very important for degradation. So you have one organelle for storage, another for degradation, and then you have transport mechanisms, and you have one transport for that is for reserpine. That was the one that we hit upon for the first time and could locate by means of different various experiments to be on the very vesicles. And then we could identify imipramine, that you have the name on the bottom there. And this is the transport mechanism for bringing back the released transmitter back to be reused by the nerve terminal. And then we could identify the receptor. The receptor, when we started with receptors, nobody was interested in receptors. They wanted to have, like the enzymes for synthesis, degradation, but we said, look at the receptors. So that was our experiment that we saw, that put the point to the receptors. And chlorpromazine acted on the receptors for, first of all, dopamine, but also to some extent for noradrenaline, very much, and to little extent, some extent also serotonin. So that is how the whole thing could be put together. And this, I think, is the first model of a central synapse.
Yeah. I've used this old picture drawn up by a collaborator of Arvid from this time. It's quite a classical picture, but by and large, it's still very relevant. Not much has happened since then. This
a lot of details have come in, but nothing is wrong.
Nothing is wrong
in it. It's still correct. Yes.
A few years later, the Americans also adopted this and called it the NIH model of the synapse. And of course, that stands for not invented here, because it was invented in Gothenburg, in fact. Okay, we'll come back to the reuptake, for example, shortly. But this is just to mention, because we will come back to this when we discuss your new drugs, that we have different dopaminergic systems in the brain. Could you briefly just tell us about it?
Yes, of course. The one that came into focus first is that has the number 1, nigrostriatal, that goes to the basal ganglia. And that is involved with movements very much, but not only movements, also, a lot of what is important for us, the way we feel and the way we think. It's so much in the big one from the black nucleus, substantia nigra, to the basal ganglia. But then there are others that go actually traveling to the cerebral cortex and different. And actually, they are also not shown here. Some go down to the spinal cord. So, I mean, the dopaminergic system is everywhere. And you have this very important tuber, the number 3, that tuber infundibula, that's so much involved in the control of our endocrine system. So dopamine is involved everywhere.
And could we say, because this is important when we discuss your new drugs, in Parkinson you want to activate the nigrostriatal neurons, and in schizophrenia you want to dampen the activity of these mesolimbocortical. Would you say that?
Absolutely.
Okay, and we will come back to this. So before, we have not too many minutes left. I don't know about the time, but 1 minute left? No, that is not, because I have the time here. We have somewhat more than that. We have 5 minutes if I'm... Okay, thank you. Thank you, Mr. Chairman. Very briefly, I will just briefly mention serotonin, but we have not time to get into this. But this is another accidental finding, how they observed that imipramine that you mentioned was a very effective antidepressant. That is before we had any treatment for depression. And then that was also in the 50s. And then you discovered, Americans have claimed that imipramine inhibits reuptake of noradrenaline, but then you pointed out that it also inhibits reuptake of serotonin. Then the Americans believed that antidepressants act by noradrenaline. You suggested serotonin. You went to a small pharmaceutical company in Gothenburg and suggested that they should develop a selective serotonin reuptake inhibitor. You did so quite rapidly, and here's the first patent patent for such a drug that is before Prozac. So Prozac is not the first SSRI. This is the first SSRI, okay? And now, as you may know, the SSRIs are first-line treatments not only for depression but for a number of other psychiatric disorders. It is used per today by 7% of the population. Many more have used it for shorter periods, so it has been used by a large number of patients. And after these drugs have been introduced, the suicide rate has declined in society considerably. Could you foresee this development when you developed the first SSRI?
No.
Not at all?
No, not really. I mean, it was an obvious hypothesis because the earlier drugs, imipramine and the others, they acted also, as we had seen, not only on noradrenaline, but also on serotonin. So it was an obvious question to ask. Yeah. How about if you just act on serotonin, what happens?
You were lucky again.
Yes, I was astonished.
Okay, 3-4 minutes more about your current thinking. First, it was the idea of a partial receptor agonist. Then we come to the stabilizers. But why did you suggest that one should treat schizophrenia? What is a partial agonist, and why should they be used for schizophrenia?
A partial agonist is doing the same thing as the endogenous agonist, which is a full agonist. Evolution has made them very powerful, but if you modify the molecule to some extent, you can make it bind to the receptor and stimulate the receptor, but not all the way up, only a bit up. That's a partial agonist. So therefore, a partial agonist can also be called a mixed agonist-antagonist, because if you have a full agonist binding to the receptor, and you have a lot of that on the receptor, if then you put in the partial agonist, you displace the full agonist, and therefore you will see that if you're coming with from a high occupancy, it will be an inhibitor. On the other hand, if there is nothing really at the outset on the receptor and the partial agonist then it stimulates. But it's do that to a certain level which is lower than the 100% level. And that's the idea there is that will be a much milder way of treating the brain. One should always try not to be like an elephant in a porcelain shop, which one easily can do when you work on the brain.
Okay. So, and this led to the introduction of aripiprazole, that is a new antipsychotic drug that acts by this mechanism that you suggested, being a bit milder in its side effects. Finally now, since it's Bengt's birthday, we must obey him, so we cannot talk too much about this. Say 2 minutes just about your recent pet project, the Dopamine Stabilization. And this is your novel drug that you hope shall revolutionize psychiatry and neurology, as I assume. And what you say here, this is Arvid's own slide, I will just read it for you. To get beyond the simplistic characterization of brain disorders in terms of either hyper or hypofunction of neurotransmitters, that was the aim of this project. Why is this concept simplistic? Because it erroneously assumed that the dysfunction of neurotransmitter is consistent, that is the same in different neural circuits or at different times. Examples showing the opposite, and we have spoken about this already, so you can skip these slides. So what you are saying here is that given this inconsistency, that some pathways may be hyperactive, some may be hypoactive, you say that any drug advancing or inhibiting the function of a given transmitter without any limitation will have adverse effects inherent in its mechanism of action, and this will seriously limit its usefulness. And you have some examples, for example, for antipsychotic drugs. So this inconsistency hurdle is overcome by your new drug. Why is this important?
It's important because a given neurotransmitter, such as dopamine, in some neural circuits, it operates normally, even at a certain doses. In others, it's high. In other parts of the neural circuit, it's low. So therefore, if you do something that works only in one direction, it will cause side effects. But our drug will is a miracle drug in the sense that it will bring down what a dopamine function in those circuits where it's high. It will bring it up in those neurocircuits where it's low. So it's a stabilizer.
And I have shown this in this slide. Slightly, not very much moving animals are stimulated, and those moving a lot are not stimulated at all, but on the contrary, inhibited. Okay, I don't think we have time to go into this. You suggest it is due to a blockade of extrasynaptic receptors, but I think we will have to skip it because Bengt seems very... I will just finish by saying that Arvid is now in the midst of clinical exploration. Your final comment will be on this slide.
Yes, this has to do with mental fatigue, which is a very, very important condition in connection with many different CNS disorders, and a very disabling condition. And in mental fatigue, we have done clinical studies, a number of clinical studies, which all show a clear effect on mental fatigue, in the sense that people who have been unable to do anything really, getting tired only after an hour or so intellectual work, and then stop doing for example, particularly after stroke. If now you give this drug, they will start to do things. Their feeling of fatigue, being tired, is very much relieved, and you have all kinds of variables that can read that improve. So we have been very successful, and we think this is going to be a very important drug in many different neurological and psychiatric disorders where fatigue is an important.
And you don't have the side effect you would get if you stimulated the system, and not those that you would have if you blocked the system.
Almost no side effects.
No side effects. With that word, I will think we thank our chairman. Thank you very much.
So there is a future. We have time for one question. Well, then I ask, why small molecule?
Up there.
Yeah. Microphone now.
Do you think that dopamine is somehow related to the placebo effect?
Please repeat it.
Do you think that dopamine is somehow related to the placebo effect?
That it's related to placebo? No. No. Next question. You there.
No, in fact, there are strong data. Is all this a placebo effect? What would you say?
Of course, we are comparing the drug with placebo, and we have very strong statistical evidence that this is not a placebo. Even placebo can work via dopamine. So it's an interesting interaction. But this is different from placebo.
Yes, one more.
When you were talking about the rabbits, you said that you were also able to decrease the amount of dopamine. But I mean, how did you do that? Because doesn't the body develop its own dopamine or produce it? How did you manage to lower the rate of dopamine being produced? That was...
I think, how do you mean how to do it, or the consequences of reducing dopamine?
No, not the consequences, but because the body produces also its own dopamine.
Do an organism survive without dopamine? Could you just take the dopamine away and still survive? As you did in the rabbits. Isn't dopamine essential for the living of the organism?
Absolutely.
But the rabbits, the serotonin you can live without.
Oh yes, of course. Wild animals will not be able to survive at all.
No.
A lab animal that's being taken care of can survive without dopamine for a couple of days, I think. But then, of course, since the animal stops eating or drinking, the survival is very short.
But some
I'm sure
transmitters are less essential for living. Serotonin you can live without.
Oh yes,
Yeah.
yes. Dopamine is so special. Well, of course there are others. I mean, if you go to the amino acids,
Yeah.
glutamate and
You would go kaput.
they will not
I'm
survive.
sure you have many questions, and there is a mechanism by which you could file your questions on a piece of paper, and we will look at it. I think one question I can tip you about is, why are these molecules so small?
Well, yes, that's an important thing. It has to do with if you compare with hormones, they can be small molecules, but they can also be big molecules. But when you come to a neurotransmitter, it can be only manufactured in the cell body with the nucleus and all those mechanisms that you have there. Then it has to be transported down to the nerve ending, and if it's a big molecule, it's kind of awkward because it has to be. You cannot manufacture it down at the place where it's going to work. So therefore, it's very smart to go to small molecules instead, because then you can just transport the enzymes that are necessary to synthesize, release, and all those that will have to be done around the cell, the nucleus. But then the manufacturing is down at the nerve terminal, and only small molecules can be used for that.
I think that's the correct answer. So thank you once more, and now it's lunch. You did well.